Welcome to my channel Adhyan with Flow. Electron transport system. Our today's topic is electron. How electron transport by their system, and we can also call it as a oxidative phosphorylation. Now, where we get electron? Look at here. In previous classes, we have studied that in the glycolysis pathway, glucose gives to pyruvate glucose 6 carbon molecule. They give us 3 carbon molecule product. Now this is the first cycle of pyruvic acid. We know now we get two pyruvate molecule from the glucose. Now this is the first pyruvate cycle. Yeah, and here another pyruvate cycle produce there another Krebs cycle. So citric acid cycle conduct into 2 times. Not a single time, conduct 2 times. This one pyruvate conduct their cycle and another pyruvate acid come and they conduct their cycle. So 2 times this cycle conduct, and this reaction happen twice because this is the one pyruvate cycle and another pyruvate, they also do same. Clear guys? So 2 into 2 into 2 into 2 into 2 into here also 2 into 2 into 2 into 2 into 2 into 2. So this cycle happened twice. Now NADH and FADH, they go towards the ETS system and participate in electron transport system. They contribute their electron. Look at here guys. Look at here. The following steps in the respiratory process are released, yes, and utilize, yes, the energy stored in NADH plus and FADH2. Look at here. They use what kind of energy? NADH2 and FADH2. Look at here. This form and this form. They utilize now. After utilizing, they free there. They also free because we take electron from them, and this NADH plus participate here. This FADH plus participate here, and they convert. And this, understand? I think now you understand where we get from electron here to here. After giving electron, after utilizing of electron, they go towards the TCA cycle and take a participate in this sequence. Now here to here. Look at here. This is accomplished when they are oxidized through the electron transport system, and the electrons passes on oxygen, resulting in the formation of H2O. Now H2O is formed. Really H2O is formed. Now really H2O is formed. This is the metabolic water. Yes, I teach everything, don't worry. I'm just reading the paragraph. The metabolic pathway through which the electron passes from one carrier to another carrier. What do you understand by carrier? We know now we have plasma membrane. Our plasma membrane contain multiple types of carrier, multiple types of channels, multiple types of pump. We know now what they do. They do molecule passes toward this to this side and this to this side. Same thing. They transport molecule, ions, minerals, glucose, amino acids, etc. Some carrier require ATP like pump, and some carrier do not require ATP. We call it as a channel and carrier also. So to another is called electron transport system. Let's start. Where this process occur? In inner mitochondrial membrane. We know now this is the outer layer of mitochondria and this is the inner layer of mitochondria. This mechanism happen in inner mitochondria. Electrons from NADH, look at here, NADH, produce in the mitochondrial matrix during, look at here, citric cycle, are oxidized by an Nadh dehydrogenase, very important enzyme, and electrons are then transferred to the ubiquinone located within the inner membrane. Look at here. Now you can understand better. They draw what they draw? They draw this to this. This is the layer of inner mitochondrial membrane. This is the inner mitochondrial membrane. Look at here, here to here. Look at here. This side touch with matrix. This side touching with matrix, and also this side here to here. This is also the side of plasma membrane. This is the also side of plasma membrane, and this side contain with intermembrane space. I'll show here. This is the mitochondria, outer layer. This is the inner layer. So this is the inner layer. Clear? This is the inner layer. This zone. Now it's clear. And now this portion, matrix indicate this portion. Here plasma membrane is karna. Here plasma membrane is karna. This portion is this. And now what is the intermembrane space? Look at here, this space. So clear? This membrane touch this side and this side. This side is here and this side is here. Now it's clear. Guys. Let's begin. So the inner layer, this inner layer contain some complexes. They contain some complexes. Now one by one we are going to study about this complexes. One by one we are going to study about these complexes. Let's start. Complex first. I'm indicating here. This is the complex first. Where they located? Inner mitochondrial membrane. Look at here. This is the complex third. This is the complex third. And this is the complex 2. Look at their arrangement. Remember their arrangement, okay? Be specific. This is the complex 4. Clear? And this is the complex 5. And we can also call it as a ATP synthase pump. ATP synthase pump. ATP synthase pump. Now clear? Now this time is clear now. This is the complex 1. This is the complex 2. This is the complex 2 here, not here. This is the complex 3. This is the complex 4. This is the complex 5. Now everything is clear. I'm removing now. Now, what happened in complex 1? Yes, we know that NADH, this thing, convert into this thing. This energy form comes from citric pathway. We can say it as a TCA. We can also say it as a oxaloacetic acid. We can also say it as a crab. Clear? This complex 1 name is NADH dehydrogenase. Remember, important. Remember every term. We get NADH plus this energy. This energy form convert into their less energy form. This energy form convert into their less energy form because they give their hydrogen. They give their electron. So first what they give? They give their hydrogen and they give their electron. They give their hydrogen, they give their electrons. Conversion of this to this process, they loss of some electrons here and they lose some hydrogen ions here. Now this NADH again go towards this cycle and they take energy and again come. This process continue. So the first complex is dehydrogenase complex. Full form is NADH dehydrogenase. Remember this thing. Where this reaction is occur? This occur in the matrix part of the mitochondria. And where hydrogen ions will move? They move towards the intermembrane space. Now, this electrons carried by FMN. And now FMN pass their electron towards the FES. This step is clear. Electron passes towards FMN and FMN passes their electron towards the FES. Now, FES passes their electron towards the ubiquinone. Look at here. This line is complete. Electron then transferred to ubiquinone located with inner membrane. This is the inner membrane. Clear, guys? So electron passes towards the ubiquinone. The full form of the UQ means ubiquinone. And ubiquinone pass their electron towards the ubiquinone H2. They passes their electron towards the what? Where? UBQH2. And UBQH2, they pass their one electron towards this side. They have two electrons now. They pass their one electron in this side and another is this side. Look at here, guys. So again we get 4 hydrogen ions. These hydrogen are very important because due to these hydrogen we get energy in the last step. So in complex third, what they do, take electron from UBQH2 and transfer these electron towards the cytochrome B. Cytochrome B transport their electron towards the FES. FES transport their electron towards the cytochrome C1. Cytochrome C1. Clear guys? And cytochrome C1 passes their electron towards the cytochrome C. And now cytochrome C passes their electron to complex 4 because complex second does not take participate. They does not take participate in this transport. They solely participate in the electron flow. They divide, they convert succinate to fumarate. They convert into succinate to fumarate. They transport their electron towards the FAD. FAD transport their electron to FES, and they pass their electron towards the ubiquinol, and ubiquinol passes the electron to UQH2, and they gives the electron. Okay guys, this complex located here, they present between the complex first and third because of it does not participate in the electron flow. So we are writing here what they do. They convert what? Succinate. This conversion occur in matrix fumarate, and due to this conversion, electron is removed. Electron is taken by FAD. FAD give their electron towards the FES, and FES give their electron to theirs. Now it's clear. The complex second name is, the name of the complex second is here, complex second succinate dehydrogenase. Actually this complex is located here between 1 and 3rd, but they does not give any kind of electron. So that's why, because they don't take any kind of electron from the complex first. Where? Look at here, complex second give the electron towards the complex 3rd, but they does not take any electron from complex 1. No. So we all writing here, they does not take complex 1, directly transfer their electron to complex 3rd, and complex second they give their electron to complex 3rd, and now they give their electron to complex 4. This is the phenomena. Now complex 3rd getting electron and transfer towards the complex 4. Look at here. Now in complex 4, copper accept their electron. They transport electron towards the cytochrome A. Cytochrome A transport their electron cytochrome A3, and again cytochrome A3 passes their electron towards the copper. Okay, so now the first water molecule is formed. This electrons taken by and made half molecule of oxygen, some hydrogen ions, and electron. They make one water molecule. Now the first water molecule formed in our whole metabolic pathway. Here, where? At the complex 4. The name of the complex 4 is Cytochrome C Oxidase. Remember their name. Now this conversion give another hydrogen ion. Look at here. Now this time this hydrogen ions, and this hydrogen ion, they are maximum in inner membrane space. Now they goes towards the ATP synthase. Now they goes towards ATP synthase. We can also call it as a, remember now, inner wall contain F1 and F node particle. Yes, so these are F node. They take their complete H plus and transport towards F1 here. This take hydrogen ion and they transport towards the F1. Here ATP synthesis work their function, and what they do, ATP to ADP conversion. They take one by one hydrogen ion. They take one by one hydrogen. First they take hydrogen and they convert ATP. Then another hydrogen is come. They make ATP. Yes, we take ATP. Now another hydrogen come, another, and they do the same process. So ATP will form. Highest ATP will form in electron transport system. So that's all about ETS. Look at here. Ubiquinol also receive reducing equivalents via FADH2 complex second, that is generated during oxidation of succinate in the citric acid cycle. We know now complex second, what they do, they change succinate to fumarate. This process is escape from the citric cycle, but succinic they change into first fumarate. This process is escape here. So here succinate converts into first fumarate, then malic. So this process occur in complex second. The reduced ubiquinol is then oxidized with the transfer of electron to cytochrome C via cytochrome Vc1 complex. What is the cytochrome Vc1 complex? This is the complex second. Look at here. Remember their name, B specific. Cytochrome C is small protein attached to outer surface of inner membrane and act on mobile carrier for transport of. Yeah, they take electron from complex second and they take electron from also complex first. Look, I'm writing here now. They take electron from complex first. They also take electron from complex second. So the transfer of electron goes towards the complex third and fourth. Complex fourth referred to as cytochrome C oxidase. Complex containing cytochrome A, yes, A, and A3, yes, A3, and 2 copper centers, yes, yes. When the electron passes from the one carrier to another via complex 1 to 4, yes, 1, the electron transfer towards third, then fourth. Look at here. Now multiple hydrogens are collect and they are coupled and they go to reach towards the ATP synthase. Look at here, ATP synthase for the production of ATP. Look at here now. Every hydrogen ion come and contribution for ATP formation. So the number of ATP molecules synthesized depend on the nature of electron donor. How much electron donors are present? How much? Oxidation of one molecule of NADH gives rise to 3 molecule of ATP. Means oxidation of one nitrogen NADH, they give 3 molecules of ATP. Means at the same time they release 4 hydrogen ion. They give from 4 hydrogen ion we get 3 ATP at a time. Why that one molecule of FADH produce 2 molecule of FAD? Means if we break NADH, they gives to us 3 ATP. If we break FADH, they gives us 2 ATP. Clear na guys? Although the aerobic process respiration takes place only in presence of oxygen, here oxygen is required. We don't use oxygen, a single molecule of oxygen in citric cycle. The role of oxygen is limited to the terminal stage of process. Yes. Here, complex 4 use half oxygen and 2 hydrogen ion and 2 electron. So yes, the presence of oxygen is vital. Yeah, necessary. It drives the whole process by removing hydrogen ion from the system. Oxygen acts as a final hydrogen acceptor. Yes, this question is also asked in examination. Choose the pick one who act as a final hydrogen acceptor. It doesn't give any other word. Choose the final hydrogen acceptor. Phosphorylation where is wait. Phosphorylation where it is the light energy that is utilized for production of proton gradient required. Here multiple protons come na, so they make proton gradient. So where this pump is work? Why they use phosphorylation? Because we know this pump is attached with inner membrane. This is the inner membrane. This is the F node. This is the F1. We call it as a ATP synthase. We know that hydrogen ions come. But without phosphorylation, this pump is not activated. Require what? Phosphorylation. So then this pump is activated. After activating of this ATP synthesis, the hydrogen ion contribute to participate and form ATP. ADP changes into ATP. So this is all about your ETS. Remember everything. What is the name of complex 1? What is the name of complex 3rd? What is the name of complex 2nd? What is the name of complex 4th and 5th? They give you options like this. This reaction occur in which complex? This reaction is occur in which complex? This reaction is occur in which complex? So you have to remember that this complex reaction is occurring complex first. They give anything. First water where form in complex 4. Which complex promotes succinate to fumarate conversion? Where to where? So you have to remember this thing. Where we get 2 hydrogen ion. Where we get 4 hydrogen ion. Which complex collect electron from ubiquinone H? Which complex receive reduced ubiquinone? So complex 3rd, they receive electron from ubiquinone. So these are question making area, one word. Look at here guys, look at here. This is what outer side, this is matrix side, and this is inner membranous. So now there is F0 particle, there is F1 particle. Hydrogen ion comes and this to this formation is occur. So here ATP formation occur. This is the inner mitochondrial membrane. So that is all about ETS. Clear guys? So that's all for now. We will meet in our next video. If still you don't subscribe my channel, then please subscribe now guys. Thank you. Thanks a lot for listening me. Keep smiling. And yes, if you revise this chart, ETS, citric and glycolysis, you will perform better. You have to remember one by one word. I told you everything that how kind of question will frame here. So you have to work on it. Thank you.